let us talk about RNAs now. So RNAs are basically sister molecules of DNA, we can say, because the basic difference between RNAs and DNAs would be in terms of the sugar they hold. So as understood that RNAs are also nucleic acid, and they also have quite similar in terms of structure. The fundamental difference is that the sugars D-ribose and deoxyribose. So deoxyribose is the sugar that is present in case of DNAs, and in case of RNAs the sugar that we have is basically D-ribose. So different sugar, that is the first difference. And the second difference is obviously in terms of the structure, that is RNAs are a single strand structure. So single strand, different sugar, and uracil instead of thymine. So this is the major thing that is different between the two nucleic acids. Rest of it is same. So here also you have a phosphate group, here also you have a sugar, that is the ribose sugar, and you have nitrogen bases. The purines are same, A and G. The pyrimidines on the other hand are C and U this time. So C and U. Now in terms of the structure of RNAs, it's also a polymer, and here also you sometimes see them in different type of structure. Sometimes they are just a loose strand floating around. Sometimes they have this loop-like structure that you are seeing here and there. Partially, some places they form this double helical structure, but it's not a complete double helix, because the same polymer extends and comes back, and while coming back it forms a double helical structure with itself, right? So that is basically a single strand only, but it is twisted upon itself, and that's why Chargaff's rule is not holding good for here, because here there is no need for this balance, because there is no need for the two strands being equivalent to each other. Moreover, RNAs serve several different purposes inside living cell, and depending on that they are named differently, and their structure could also be a little bit different from each other. For example, messenger RNA, which are abbreviated as mRNAs, they provide genetic information. They are the major contributors in biosynthesis of proteins, that is from DNA to proteins. When it is getting synthesized, the step in between passes through mRNAs. So we'll talk about this. There are other RNA structures as well, like transfer RNA. Transfer RNA transfers amino acids to mRNA during protein biosynthesis. Also another very important RNA, tRNA. Along with that, you have heterogeneous nuclear RNA, ribosomal RNA, and small RNAs also. Small nuclear RNAs, small nucleolar RNAs, transfer messenger RNAs. Mostly they are found in bacteria and so on. So specific types of RNAs, which are sometimes also called riboenzymes, can also function as enzymes, and these functions either are like catalysis or assisting some self-processing reactions of RNA and so on. So they perform several different tasks inside living cells, and accordingly they can have a little bit different structures also. But in general the structure is like that they have a different sugar, they have uracil instead of thymine, and they have a single strand. So in this particular case, the bases would be adenine, guanine, cytosine, and uracil, and the nucleotides, which are now called ribonucleotides, their names would be adenosine 5' monophosphate, guanosine 5' monophosphate, cytidine 5' monophosphate, and uridine 5' monophosphate. So abbreviations would be AMP, GMP, CMP, and UMP. The D is missing because the sugar is different. It's not a deoxyribose nucleic acid. That's why the D is missing. Now let us talk about the protein biosynthesis, because gradually we'll move towards how the protein looks like, because that is also another polymer that performs most of the important tasks in living cells. So the information constituting the genome of a particular organism, that is the DNA, that is basically a sequence of bases. We saw that each of these monomers of this DNA are different only in terms of what bases are sitting on the strand, right? So that information it transcribed into RNA, which is a mRNA, and that is then translated into protein. So there are two steps in this process, protein synthesis, protein biosynthesis, two steps. First step is called transcription, where from DNA RNAs are made. So this is the transcription step, where from DNA RNAs are made. That happens inside nucleus. And the next step is called translation, where from RNA proteins are made, and that happens inside the cytoplasm of the cell. So after creating the mRNA comes out of the nucleus, and inside cytoplasm it happens. There ribosomes, you know about ribosomes, they are also a cellular component that helps in production of this proteins, right? So let us start talking about this protein. One of the very common protein, I have given you example of this protein several times, is hemoglobin. So proteins are basically polymers which have arrived into a nice packed 3D structure, which is responsible, the structure is responsible for the particular tasks they do. So protein comprises of the largest percentage of organic molecules in body. So whatever organic molecules you find in a living cell, a huge percentage are basically proteins. So the term protein, it has a Greek origin, and they are basically nitrogen containing very large molecules. We can call them macromolecules, and there are 20 amino acids in general, mostly 20 amino acids. They basically are arranged in several different ways, several different fashion, to give rise to different proteins. So the structural function of proteins can be different. Like hemoglobin, it helps in binding oxygen. There is keratin that is responsible for our hair and our nails, like keratin. Collagen, you know, it's important for bones. Different proteins have different particular functions. There are different dynamic functions also, like blood clotting factors, enzymes, the hormones, how they are working, antibodies, how they are working, membranes of cellular membranes, how they are working. So all these things are handled by proteins. So let us look at these proteins, and let us look at their basic units, that is their monomers, which are amino acids. So before that, we will focus on how these proteins are made, that is transcription and translation. So what we understand here is that inside DNA, in terms of this ATGC code, some words are written, right? And that part which is understandable by the cellular matrix or the cellular functionality is called genes. So all of DNA is not genes. So some parts of DNA where something informative is written in terms of this ATGC code, that part is called a gene, right? So this words, if you want to say, in this language, in this alphabet of ATGC, it makes sense only if a protein could be made from there. And a protein could be only made if, because proteins are nothing but sequence of amino acids, so protein could only be made if you can understand the existence or creation of a particular amino acid from a portion of DNA. So how this amino acid sequence can be understood from DNA, that is what we will focus on in today's discussion. So suppose you have a DNA molecule, right? So this is a DNA molecule and it has certain genes. So this is, say, gene 1, this is gene 2. In between, these portions are meaningless, they don't mean anything. There are separate genes, and from each gene a particular protein can be made. Okay, a particular protein can be made. How can it be made? So in the DNA strand, you know that A, T, G, C are sitting side by side in a particular order. So in this particular order, a DNA strand is there. Suppose one of the DNA strands I am considering, any one of the DNA strand you consider. Say in that particular DNA strand, this is the sequence that you are seeing right now. Okay. So in transcription step, what will happen? In transcription step, the DNA will open up. The DNA will open up, and one mRNA will be made, which is just the other half of the DNA strand, right? So suppose you have ATGC is your code, right? And instead of T you have U in case of RNAs. Always remember that. So wherever you have C in the DNA strand, you create an RNA and there you put a G. C, G, G, C, G, C, C, G, right? So whenever you have a C, you put the other half, the counterpart of that particular nucleotide. If you have A, then its counterpart in terms of DNA would have been T, but it is an RNA that you are making, that's why you put U. So whenever you have A, you put U, right? Whenever you have T, you put A, and that is how you create the first step of transcription. You create an RNA. This is an mRNA. Once the mRNA is made, the next step is called translation, and it is done by ribosomes. So ribosome is a small nano machine that lies inside cell. What ribosome does is that ribosome comes and sits on this RNA, and it can move, like considering it as a rail line, as a railway track, as if the RNA moves on top of this and starts reading this in terms of a 3 nucleotide code. So side by side, if it finds any 3 nucleotides, say UUU, that means something, right? GGC, that means something. What does it mean? It means a particular amino acid. So when ribosome will sit and read this codon and understand that this particular code means a particular amino acid, it will ask transfer RNA, tRNA, to bring that amino acid. Transfer RNA will bring that amino acid, and that will be stitched here with the ribosome. Then the ribosome will move on and read the next part, 3 codon, next 3 nucleotides, and from there it will see GGC, that means something, that means a different amino acid. We will come to that how it reads, what is the exact code means. So it reads a certain amino acid, transfer RNA brings and stitches that amino acid with the first one, and this way the first part of the protein starts getting made. This is a polymer being made, right? So amino acids in general, in human beings, most of the living organisms that we see around, amino acids, 20 amino acids are abundantly found. So you can consider this to be a long chain where 20 different monomers can come in any order depending on how the RNA reads, how the RNA is read by the ribosome, right? So this is what is getting made is called a polypeptide. It's an amino acid chain, and when it is only a few amino acids, it's called a polypeptide. When this polypeptide is long enough and it is detached from the ribosome and it can orient itself and get its 3D conformation, then it is called a functional protein, right? So this is how the proteins are made. So the most common amino acids are this. So this is a list, and this list is something that is always going to be associated with this understanding of how these proteins are made. So alanine, arginine, asparagine, aspartic acid, all these different amino acids are there, and these are their 1-letter codes, right? So 20 amino acids. Now let us understand the genetic code, that how each of these serial 3 codons means something. So this is the table for understanding it. If you understand this table once, then you will not forget it, and you will be able to see if you have a DNA part and I ask you that what protein is going to be made from here, what is the polypeptide chain that is going to be made there, then you will be able to do that. So first, if you have a DNA chain, create the corresponding RNA chain. Once you have the RNA chain, then you start reading them 3 alphabets at a time. So this side of this table is the first base. That means if you have U, then the first base U is here, first base C is here, A is here, G is here. First base means when you have this RNA, what is this first letter you have here, right? Next codon, first letter G. Next codon, first letter C. Next codon, first letter U. So the first letter you will be able to find here. So track your first letter. Then the second letter, second base is here. So second base you will find here U here. So UU here, UU, all the UUs are here, right? CUs are here, AUs are here. So this is for the second base, and then the third base is here, right? So UUU will be the first one, UUC will be the second one, UUA will be the third one, UUG will be the fourth one. Okay, so this is how the genetic code is read, and for each of this combination what particular amino acid is meant is written here, right? This is the 3-letter code of the amino acids that is written here. So once you understand this table, you will be able to decode any DNA in terms of a polypeptide, and you will be able to see that how this biopolymer is made, right? So I will give you certain exercises on this so that you can work on this particular thing. Try to understand this table by looking into it thoroughly. In this particular table, there are certain sequences that mean start or stop. For example, AUG, it is something that if it is found somewhere, then it will start translating it, right? And when you find three particular sequences, UAA, UAG and UGA, these 3 sequences mean stop. So whenever you find this, you stop. So it looks like a very cryptic message is written inside DNA and the ribosome is trying to read it. So start is AUG, stop is UAA, UAG and UGA, right? Rest of them are some or other amino acid. So you read them in three letters and you create the polypeptide chain. So if you want to see here, let us do a quick exercise. Suppose this is the DNA and this blue one is the transcribed strand. That means that from that strand RNA is being made, right? So let us look at the strand. It is having T, A, C, T, T and so on. So wherever you have T, you put A. Wherever you have A, that means here you had T, right? So wherever you have A, here you cannot have T because it's a RNA, you will have U, okay? Then C, counterpart is G, T, counterpart is A, T, counterpart is A and so on. It goes on. You create this mRNA strand. Once you create the mRNA strand, the transcription step is done. Next is translation. So for translation, three codes you select. So first one is AUG, next one is AAG, third one is UUU and last one is UAG, right? So AUG, let us go back and check what is the genetic code. AUG, A is here, U is here and G is here. AUG means start. So whenever it is the first codon, you don't have to write anything. You can just start translating it. Next is AAG. Let us go here. AAG. AAG is A first letter, A second letter, G. So AAG is here less. So they have written less, right? So MET, less. MET is the start codon. Whenever you have AUG, you start it with MET, right? Here it is written, start it with MET, start and then less. What is the third? UUU. UUU. Let us find it. UUU, the first one. It is PHE. So let us write PHE, right? And the fourth one is UAG. So UAG is stop codon, so you stop, right? So this is how you make the polypeptide chain. I hope you understood how to create this polypeptide chain. Now, interestingly, what happens is that sometimes mutations happen in DNA sequences, right? Mutation happens causing error during DNA replication, or maybe while mRNA creation, something got changed. And from there, some genetic diseases can get created. For example, sickle cell anemia is one of the most common anemias that happens due to hemoglobin, a wrongly made hemoglobin. And that happens because in a normal hemoglobin, the sequence in the DNA is CTT, that is needed to be transcribed into RNA, right? But in case of mutant hemoglobin, this T is replaced by A, and thus this be replaced by U, and thus instead of the normal hemoglobin, one particular amino acid changes to another particular amino acid, right? It has to be GAA. From there you should have GLU, but instead of that you have VAL. Once you have VAL, this is basically the sickle cell hemoglobin. That is how the genetic diseases creep in due to the existence of mutations. There could be different kind of mutations. For example, suppose you have a normal gene and it might happen that a base is substituted. That means instead of G you have A. That happened in case of sickle cell also, right? You replace something by something else, creating a new amino acid in the sequence. Instead of gly, here Sir is made, and that's why it's a new protein. It's not the same protein as it is supposed to be, it's supposed to behave. It changes its behavior even if a single amino acid is changed. On the other hand, there could be another type of mutation which is called base deletion. That means one particular base is deleted. If it is deleted, then what happens is from that point onwards, the entire sequence changes because it's a 3-3 codon sequence, right? So if you delete this particular U, then instead of this UUU, you have UUG. And the next protein also changes to GCG. And the third protein changes to CAU, right? Complete sequence changes, and that is also a huge mistake in case of transcription which happens, and from there you result into a wrongfully made protein, right? So chromosomal changes can be small or large, and for that you can have different kind of mutations happening, and for that different genetic diseases might creep in, right? So let us stop here for today. We will talk about the structural details of proteins in the next lecture.